Okay, thanks very much, everyone. My name is David Furley. And thanks very much for coming out to the Haymarket Theater tonight. This collaborative production of the Marshfield and Plainfield Conservation Commissions and the Cutler Memorial Library in Plainfield, the Jesuit Public Library in Marshfield, Goddard College, and Natural Marshfield. This event has been also made possible by the support of Bear Pond Books, WGDR, Goddard College Community Radio, Kellogg Hubbard Library in Montpelier, and Central Vermont Community Television. The donations at the door, we thank you very much, will go towards the next event, perhaps in the spring and hopefully for years to come. David Montgomery is an internationally recognized geologist who studies landscape evolution and the effects of geological processes on ecological systems and human societies. Author of 3 award-winning popular science books, he's been featured in documentary films, network and cable news, and other media. When not writing or doing geology, he plays guitar in the band Big Dirt. Anne Biklé has over 2 decades of professional experience in field biology, watershed restoration, environmental planning, and public health. The Hidden Half of Nature is her first book. Anne and David are married and they live in Seattle with their black lab guide dog dropout, Loki. After Anne and David's presentation, there will be a very brief Q&A ending with a longer book signing. Thanks very much, and please welcome David Montgomery and Anne Biklé.
All right, well, thank you for the introduction. I'm obviously Dave, and Anne, my co-author, is over in the corner. How this is going to work is that I'm going to start the talk and then hand it over to her about halfway through. Because as you'll see, this book was really very much a collaboration. She's a biologist, I'm a geologist. And we actually ended up writing about microbiology, neither of our specialties. So how did we actually come to write a book about something that neither one of us are acknowledged experts in? It's in part, that is in part the story that we're going to tell and the story that we tell in the book. That relates our journey of discovery of just how important the world of microbes are to the world of nature that we know. And we really are, you'll see how we got into that as this unfolds. But what we really learned in writing this is that we're living through a legitimate scientific revolution in the way that we see the invisible part of nature, the hidden half of nature, the microbial world, the life that's too small to see, and the life below ground that's also sort of hidden from our indirect senses. You don't get to live through too many scientific revolutions. The last one in my field of geology legitimately was really the idea of plate tectonics that by the time they made it to undergrad school had all been played out before. So when we started looking into this world of microbial ecology and biology and realized just how much our view of that part of nature is changing and has changed since we were in graduate school, we got really excited about it and really went down the rabbit hole trying to figure out what was happening and how to actually understand it ourselves so we could relate it to others who are not specialists in microbiology. That's essentially the preamble for how we got into looking at this. We just think it's a very exciting field, and we were glad to try and dip into it to further our own understanding. So let's start with basically looking at who are the actors in the microbial world, whether it's the archaean and the bacteria. A century ago, these would have been both lumped into bacteria. We now know that they're actually very different. There's fungi, protists, unicellular organisms that have a well-defined nucleus. We'll get back to that a little later. The viruses that biologists keep arguing, are they alive, are they dead, what actually are they? Were they once alive, are they retrograde life? But these are our basic cast of characters. And if you're interested in why fungi, which some of you may know as mushrooms as part of fungi, are actually included in the microbial world, well, we were curious about that too. We have a footnote that goes into explaining why biologists really look at it that way. But the basic answer is that their cell walls aren't separated from each other. There's holes that go all the way through. We go into that. That's not the primary thing that we want to lead with here. want to basically look at is the title of the book, The Hidden Half of Nature, is meant to be taken literally. If we look at the world of nature that we know, the kind of natural history people like Anne and I are, what we studied is essentially just half of it. It's the visible part, the part we can see with our own senses. So if we take the world of nature, ranging from down to sort of the basic building blocks of DNA up to people, and we look at the size of things, just going from 1 nanometer scale down here at the left up to meters over there on the right, and each one of these steps is a jump in size by a factor of 10. The visible-invisible world boundary is right about in here, about the size of our own red blood cells. Those are sort of the giants of the microscopic world, and they're giving rise to amoebas on the fine scale of the visible world. And you'll notice that there's 5 cycles of 10 here, 5 cycles of 10 there. There's as much range in the size of the world of nature in the invisible part of, in the microscopic part of the world, as there is in the visible part. There's an incredible diversity of sizes and ranges, and if you actually look at weighing up all the microscopic life on Earth, the visible and the invisible range of the spectrum, they would weigh about as much as all the nature that we know. Think about that. All the microbes on Earth weigh about as much as all the plants, animals, people, everything that we know of in nature. Fully half of nature's in this invisible realm. And if you took all the microbial life on this planet, some 10 to the 30th, the 1 followed by 30 zeros, a nonillion of microbes, individually, they're really, really small. But if you stretched them all end to end, they would reach Alpha Centauri and back. So they could beat us off this planet if they got organized. And held hands all the way there and back. So how did people like Anne and I, a field biologist and a field geologist, get into writing about the world of microbial life? We did it by buying a house in North Seattle. We're just visiting this region for this fall. We actually teach at the University of Washington, and we bought a house in North Seattle after I got tenure, and we settled in there. This was the house that we bought. You'll notice this lawn. This was not the best yard that my gardener wife desired. This is what the house came with. And her dream was to actually turn this ratty lawn, this 90-year-old old-growth Seattle lawn, into a true garden. And when we bought it, she viewed it as a blank slate. This sort of a labor of love started to unfold. But what happened is that after we got to peeling that lawn back off to try and actually create that garden, we found what is really the bane of most Seattle gardeners' existence. Our lot was developed on glacial till. There was not much organic matter. We didn't find a single living worm in that soil when we pulled that lawn off. And I kid you not, there was no macroscopic life, no visible life. I'm sure there are a few microbes in it, but there probably weren't even many of those. It was essentially a biological desert, and it looked essentially like beach sand, sort of a khaki color, and it's basically pieces of Canada that got left in the Puget Sound area by a glacier that then was overridden by a mile-high wall of ice, about 3 times as high as the Space Needle, if you want a reference for it, compressed into what we like to call nature's concrete. Not really the best soil to try and grow anything in. And this bothered Anne, it bothered me as well, but she was the gardener, but it bothered her a lot more. And she decided to try and take matters into her own hands and try and change the soil. Because if you look at what makes for fertile soil, it's the marriage of geology and biology, and that's why Anne and I can write about this kind of stuff. It overlaps our areas of interest. And there's a mineral component to it, and there's a biological component to it. And in that tilled soil, we had the geology, but we didn't have the biology. And so what she started to do is essentially add organic matter. She would gather the neighbor's oak leaves in the fall, and they were just more than happy to have her break them up, take them back to our yard, and dump them in our planting beds. Arborists would when they were chipping things in the neighborhood, we'd let them dump them in our yard. So we could provide compost and mulch and try and get organic matter back into our yard. And at the time, we were doing it to essentially try and keep moisture in the ground to help things grow, because we started planting in August, which is not the smartest time in Seattle to plant. We get plenty of rain, but not in August. And what we actually started to do is to also plant things in the planting beds. So we were adding the brown goods of essentially leaves and wood chips, carbon-rich sources of organic matter, but we were also planting nitrogen-rich sources of green organic matter that could be left rot and go back into the soil. So we're trying to amend the soil with sort of the very old-fashioned methods of mulching, growing cover crops, and composting. We got lots of coffee grounds from all the coffee shops that are ubiquitous on the corners of Seattle. They love it. They'll put them all, they'll put their used coffee grounds out with the garbage, and gardeners flock to grab them and bring them back to their yards. So we put a lot of composted coffee grounds, started composting our kitchen scraps, and that was other sources of organic matter we added to the yard. And I started doing something that at the start, that, to be honest, I thought was really not going to work. I started wondering, why would this work? She came home from the Seattle Flower and Garden Show, I believe it was, or the home show, I forget which, with a compost tea brewing mix. And what that is essentially it's a way to brew up a lot of microbes. You get a 5-gallon bucket and an aerator, just like you would use in an aquarium, to pump oxygen to the mixture, and you seed it with an inoculant of microbes, just like you were making sourdough bread, say, and you put in a source of sugar, something for the microbes to eat, so they could reproduce and thrive and multiply. So you're brewing up a microbial tea, which she then sprayed on the plants, sprayed on the soil, sprayed on everything. And I was deeply skeptical. I mean, I'm a geologist, and I was like, what's, spraying all these invisible life forms on the earth, what is this really going to do? And I've never been so happy to be so wrong in all my life. That cuts to what eventually happened. But after about 5 years of her intensive efforts to add organic matter back to the yard, what we in the book call her organic matter crusade, we started to notice changes in our soil. And you remember that picture of the soil that I showed you at the start, where it was essentially like beach sand. This is a pit that was dug about 5-6 years after she started with her organic matter additions to the yard, and that tan glacial till is still down there below the surface, but up at the top you'll notice the compost, the wood chips that she has at the top, and there's about 2 inches of actually fairly decent soil that has started to form in about 5 years. And you notice the plant roots go down to that layer, and then they go sideways, because they're not going down into that glacial till. Well, at the time that this was all happening, I was writing a book that's over here called Dirt, the Erosion of Civilizations, that looked back at how the archaeological record of soil erosion in ancient civilizations demonstrated that civilizations that destroyed their soil took out the source of their own prosperity and longevity. And you can actually predict the lifespan of human societies based on how they treat their soil. And I was writing this book as an academic exercise. I was interested in it as part of my job at the University of Washington. But the bottom line message that farming practices over many millennia, particularly the over-reliance on the plow in environments where it wasn't necessarily the best thing to rely on, had degraded soils for society after society. Anne was showing the way around to turn that around right under my nose out in the yard. Because if you look at the rate that soils are actually formed naturally in nature, you can't get 2 inches in 5 years. You're measuring inches in centuries. This is orders of magnitude faster than you would expect nature to make fresh soil on her own. And that got us both thinking, well, what is it? What's actually driving this? How is that organic matter that's being added to the surface of the soil being so rapidly converted into fertile soil that within a few years is actually supporting an explosion of life above ground in the yard and transform our own daily experience of our own environment? And I don't know if you recall the photograph that I showed of the lawn at the start was stripped off, but this is the neighbor's house in the back that was so bare and exposed. Now we can barely even see them. And they're perfectly nice people, but we prefer not to see them on a daily basis. This is one of the disadvantages of living within a major city. You really can get to know your neighbors too well unless you plant a lot of stuff between you and them. So we started to realize that restoring the soil in our yard had led to an explosion of life above ground. That was the foundation for essentially regenerating the garden and the yard. And also planting beds to try and grow some food outside our back door in our yard. That got us thinking about the connection between how you influence the soil, what that does in terms of the food that we eat. We actually sent some of the kale that she was growing away for chemical analysis and compared that to the average of what you would find in a grocery store. And it was much more nutrient dense, much more nutritious. And this all started us thinking, what was happening? Well, one of the big differences in the yard was that the carbon content went way up. Under our lawn, after we finally got around to testing that, as when we were just done with the book, we started off with about a 2% carbon content in the yard, 1.5 to 2%. Not much, that was that very light stuff. By the time we got to testing it about 15 years later, we built it up to about 4 or 5% under the lawn. The planting beds were up to 12 to 15%. That's as high as you got in the most fertile native prairies of the American Midwest that were so productive agriculturally. So we're demonstrating in this urban lot that you can actually regenerate fertile soil at a pace that really outdoes nature at her own game. And you notice that the kind of soil life in the yard that came back that most gardeners would recognize, these sort of cast of familiar characters here, they came in an order that we'll refer back to in a few minutes, but the part that we really couldn't see, the invisible part, the microbial part, was essentially the foundation that was starting it all. And we think that organic matter that got added back to the yard was what really kick-started the explosion of life below ground that kicked off a nutrient spiral that ended up playing out above ground in the explosion of life that we saw in the yard. And how does this work? All that organic matter that got added to the yard was being essentially ripped apart by the shredders, the kind of macroscopic life that we just had on up on the screen a minute ago. And they break it down into smaller and smaller pieces until eventually things got consumed by microbes. Bacteria, there's a clump of them over here, and fungi over here. These guys sitting by a root hair of a plant where you've got the fungi and a little hyphae sticking off into it. This is the smallest scale, where essentially all that dead matter is being recycled into its constituent components that could be used to rebuild higher order life forms. And these nematodes and microarthropods that actually graze on these finest guys, you can think about them as livestock that lives in the soil. Because what do they do with this all these nutrient-rich microbes that they consume? Well, they consume them and then they excrete them as waste. What that basically is micromanure. It's the same kind of thing. They do to the soil the same kind of thing a cow would do on the surface in terms of manuring it, but they do it within the soil on a microscopic level. And that's actually one of the main processes by which the carbon content of soil actually gets increased and improved over time. It's the biological cycling. It's the dead microbes which were eating the organic matter. So the organic matter itself that we were adding was not what fertilized the soil. It's what the microbes did with it. You could think of the organic matters feeding the invisible half of the soil food chain. And that essentially ended up adding nutrients back to the above ground half that we were after in terms of our garden in the first place. So if you think about how a plant obtains its nutrients, it obtains carbon from the air, water from the soil, mixes them up through photosynthesis, and mineral elements ultimately all come out of weathered rocks. But the organic matter, once living matter, is very rich in all the elements that it takes to support new life for the simple reason that it was alive before. And the recycling of their organic matter, the spinning of essentially the wheel of life through the living matter to organic matter and reincorporating back into living matter is fostered and driven primarily by microbial life. And you can, in great part, think of why does it take nature so long to make soils from scratch? Because rocks break down really slowly. But if you start with the biologic end of it, you can if you have the mineral end and you just add into biology, what's the lifespan of a microbe? About 20 minutes or so. You can go through a lot of generations really fast with an organism that only lives for a fraction of an hour. And so the idea of essentially increasing soil fertility through this organic matter recycling is really sort of the key to restoring soil fairly rapidly. And this led us to looking at the zone of life around the root zone of plants called the rhizosphere. It's just the zone around plants. And it's one of the most life-rich zones on the planet. If you look at the density of organisms per, say, square centimeter, or even per square meter, the root zone of a plant is off the charts relative to most other environments. We tend to think of something like the Amazon as really life-rich. Well, it doesn't hold anything compared to the root zone of a plant in terms of just numbers of organisms. And a lot of it's within just a millimeter or a centimeter of the tips of roots and root hairs. What's going on there? Well, when I was in graduate school, I was essentially taught that the roots of plants served as straws that sucked up water and nutrients to feed plants. And that's not wrong. They do that. If you crimp the stem of a plant, it doesn't do very well in getting those nutrients things up. But roots are actually a two-way street. And there's been a lot of research in the last 20 years or so that has showed that plants, when they photosynthesize, they'll take about 30 to 40% of all the matter that they're creating out of sunlight, water, and carbon dioxide, and they push it out their roots into the soil. Why would they do that? It seems pretty wasteful. They have this wonderful trick, this monopoly on creating organic matter out of raw ingredients that they get for free, basically. And they're really just going to put 30, 40% of their gross production out on the street. Well, what they're doing is they're feeding the microbes that live in the rhizosphere. And what they're doing is they're recruiting very specialized microbes, very particular microbes, that produce things that help the plants in return. If they were putting all these sugars and proteins out into the soil to feed pathogens, it wouldn't have much evolutionary success. That'd be a quick route to a short life. And yet, you look at most plants and they're doing this. Very counterintuitively to the way I was taught to think about roots as essentially a two-way street. So what's going on? Well, if you blow up the root zone around a plant, and you look at the rhizosphere, this life-rich zone around the tip of a root, it's essentially a biological bazaar where microbes and plants are trading nutrients, metabolites, and exudates. What the plants push out of the roots are called exudates because they're exuded from the roots. And those sugars that the plants are pushing out the roots don't make it very far. That life-rich zone, they're consumed, they're eaten by the microbes, and the microbes in turn push out their own waste products, their metabolites, and those include things like plant growth promoting hormones. The microbes are making plant hormones out of the sugars that the plants feed them. In other words, the plants have outsourced, have offshored some of their chemical factories, their production facilities for things that help plants grow. And some of the fungi, for example, will send their hyphae way out into the soil, and they'll prospect things like phosphorus from over on that far wall at this scale, bring it back in and trade it to the plant for sugar. So the plant's feeding the mycorrhizal fungi, and the fungi isn't feeding the plant what it needs for energy, it's feeding the plant the micronutrients it needs to make complex molecules that actually benefit its own health. Plant defensive compounds, or phosphorus, which it needs to combine with the sugars that it makes through photosynthesis. In other words, there's all these symbioses going on in the root zone of plants that benefit both the microbes and the plants. And this is a really different way to think about microbes than most of us have for most of the last century and a half. That the microbes are actually helping the health of plants. And Anne will get more into that in a little bit. I've just got a few more things to say. One of the things is that if you think about these interactions between microbes in the rhizosphere and plants, there's some really striking things that are happening. Like when insect herbivores start nibbling on a plant, that plant can send out exudates into the soil that recruit particular microbes that make particular metabolites that are taken back up by the plant that make the plant taste bad to that particular insect. This is really specialized stuff, folks. This is a lot like sort of the pollinator-plant relationships that we see above ground, but most of this stuff was unknown to us until very recently. Some of it was hypothesized, but the mechanics, the connections, how it works, were not really known until gene sequencing technology opened up the world of microbial ecology. Because imagine studying the ecology of organisms that are invisible and all kind of look the same and don't really have much in the way of appendages. How do you actually look at them? You look at them through gene sequencing. And that has resulted in an explosion of knowledge about just what's happening in these dynamic zones like the root zone of plants. And what things like Anne and I, what people like Anne and I learned in graduate school just a couple decades ago are now kind of like old news, and there's this whole new way of looking at things. And one of the more striking things that we noticed in our yard was that if you look at the order in which life came back to our yard, it was the same as the order in which life evolved on Earth. The diagram over here on the left shows you from about 500 million years ago when bacteria and fungi first came ashore in the Cambrian period, the order in which life forms evolved on Earth right through mammals and birds. That's the same order in which life came back to our yard. Of course, it happened in our yard over a much shorter time frame. It happened over the course of about a decade and a half. But why would these patterns be similar? And you may notice there'd be one big difference. We didn't get dinosaurs back in our yard. unless, of course, you count the crows, which are probably the descendants of dinosaurs. So that might even work. But why would these patterns in the small scale of the restoration of an urban lot be the same as the pattern in which life evolved on Earth? And I think it's what it's telling us is that nature's systems have been assembled from the bottom up. From the microbial scale, it's the foundation that is supporting the higher levels of organization within terrestrial ecosystems. And we've probably known this for a while, we haven't really recognized it in our agricultural and medical practices. And the last point that I'll make before turning this over to Anne for the second half of the talk is that one of the things that was fascinating to us when we started looking into this, the role of microbial life in Earth history, is the work of Lynn Margulis, one of the most interesting and I think long overshadowed scientists from the 21st century, who back in the 1960s proposed the then very radical idea that's now in all the biology textbooks, that the complex life forms that we know as animals, fungi, and plants, or the world of nature that we can study in the natural history disciplines, evolved out of the mergers of microscopic life forms. And by mergers, we truly mean mergers. What she proposed was that the first merger that led to complex life was the merger of an archaean and a swimming bacteria, where one tried to consume the other, and the impossible happened. They both survived. They became a symbiotic life form. And that happened a couple billion years ago, and was the origin of nucleated cells. And then the second merger happened about almost a billion years ago, when the product of that first merger consumed an oxygen-breathing bacteria, and again, they both survived in a symbiotic relationship, and that the ancestors of that oxygen-breathing bacterium are the mitochondria that power every cell in your body, every plant, every fungal cell, is the powerhouses of life. The third merger, of course, was when that second merger consumed a photosynthetic bacterium, the forerunner of the chloroplasts that power plants and allow them to power photosynthesis. So the subtitle of our book, The Microbial Roots of Life and Health, is also meant to be taken literally. Because if you trace the roots of life forms that we know, and trace them back to their microbial origins, it was the symbioses of individual microbes that set the pattern for the growth and diversification of all the higher order life forms. But it also turns out that symbioses between microbial communities and higher life forms greatly influence the health of life forms. And that's where I'm going to turn it over to Anne and let her take over the second part of the talk.
Okay, thank you. Get the audio figured out here. So I think you can probably tell that we were pretty enthralled, pretty thrilled with the microbial world and what it had done for us. We had put some elbow grease in for sure to restore our soil, but it was the microbes that were really doing the heavy lifting. And then, as happens with life, we encountered a big, fat plot twist. Me in particular. I had gone for a routine physical and found out a short time later from my doc that I had a cancer, that it was malignant, and that of all things, it was caused by a microbe, the human papillomavirus. I had had cervical cancer. I am happy to report today that that is behind me. I'm healed, cancer-free, but let me tell you, that was some pause for thought. It was a lot of thinking that we did, and for me personally, I began to ask a lot of questions about my health. Where did it come from? What do I do when it's threatened like that? And it coincided with the research that we were doing for the book. We were partway through writing the book when all of this hit, and the human microbiome was just starting to hit popular media. It had been in scientific journals for some time, and this just shows you what a burgeoning explosive field this is. 2014, lots of publications using that term, and it hasn't stopped, and it's 2016. So our microbiome, as many of you know in this room, It's something pretty simple, and it's surprising to me in some ways that it eluded us for so long. All it is, it's the indigenous microbial communities that live on and in us. And we thought we were so special and different from all other species that we couldn't possibly have microbes be that intimate with us. But scientists got onto this, and they began publishing it. And the few other key factoids about our microbiome. Most of our microbiome helps us far more than it harms us. We acquire it, the minute we come out of our moms, and we continue to build on that through early stages of our life. I'm thinking maybe till around 5 years of age or so. And by that time, you've been out in the world enough, exposed to enough things, you've built up most of your microbiome. And this is also interesting. If you were to count all of the cells of your body, like your liver cell, kidney, skin cells, and all of that, put them in one pile. And then if you were to count up your microbial cells, because for sure we do have them, and put them in another pile, there is at least a 1-to-1 count there, one of our cells for every one microbial cell, and estimates are putting the microbial cells up to at least 3 times that amount. And so clearly we're at least equal with them on cells, if not outnumbered. But when it comes to our genes, this is even more sort of astonishing. Through the Human Genome Project, we know that humans have about 23,000 genes that code for proteins. Well, our microbes, just the bacterial portion of our microbiome, add somewhere around 2 million more genes to our genome. If you throw in viruses and fungi, sort of the weirdo archaea, you're looking at a microbial contribution to our genome somewhere in the range of 4 to 6 million more genes. And so for me in particular, I began to think, wow, this probably has a lot to do with health. My health, Dave's health, everybody's health. And it led this sort of quest to figure all of this out. And we had done research on the plant world, and obviously, to us anyway, all of the stuff that's going on in the rhizosphere, that is driven by the microbial world. And we couldn't be that different from a plant. So this information and research that we were doing, it was falling on some very receptive ears. And it led, of all places, to the least loved part of the human body, to this fantastic ecosystem that we have on board, the human colon. Forget the Amazon, forget the Serengeti. We are carrying around some really cool stuff right within our bodies. And I want to, the next series of slides, I'm going to talk a little bit about exactly what's going on in this ecosystem, but a few key terms, okay? So if you were to do a cross-section through your whole colon, that central area is just called the lumen. And all of us, in theory, you want a nice thick layer of mucus lining your colon. It keeps things moving. It also turns out that mucus, which is produced by our colon cells, contains some exudates. Our colon cells can produce exudates. And there's theories that a lot of these exudates are feeding members of our microbiome. Our microbiome lives in this mucus and also in these features called colon crypt. And this is a little bit, this is hypothetical at this point because all the immunologists and the gastroenterologists have thought for years and years and years that this is like a force field. This layer of mucus is so thick and impenetrable, there is no way that any microbes are going to be able to penetrate that and start living so close to all of these cells. And the other reason they thought that is because all of this tissue that is on the outside of the colon wall is rich in immune cells. The lion's share of our immune system is wrapped around our digestive tract, and of that, most of it is around our colons. And this is just a blow-up of the colonic crypt, and here's a few of these immune cells. We're going to go on now to show you up close what's going on here. So this is a cross-section through the colon wall. We have that lumen area here. This is that layer of mucus. You've got your microbes living in there. Layer of mucus here. These are the cells lining the colon. And here, perched right outside all of our colon walls, are immune cells. There's a bunch of different kinds of immune cells. I want to focus on these particular ones called the dendritic cell. Now these are marvelous, marvelous cells. You can see what's going on here with this picture. Check this out. This dendritic cell cruises around the outside of your colon every now and then, actually probably more than every now and then, quite frequently, let's say. It can sort of do a little shape-shifting, and it can slide an arm right up in between your colon cells. It's probably likely doing that, at least some of us right now, I'm sure. And why is it doing that? Because it's on a fishing expedition. It lives out here, but it wants to know what's going on inside this colon, what's going on inside the lumen, who's living here in the mucus. And so our immune system is very snoopy, it's very nosy, it wants to know things. And why? Why does it want to know these things? Because it has some pals called T cells. So the dendritic cell has captured molecular samples from what's living up there in the lumen and in the mucus. And that sample, if anyone's ever been to an allergist, that sample is called antigen. It's this molecular sample, and it's very, very specific. So that dendritic cell shares it with the T cell. There's receptors on the outside of the T cell, and each receptor is made for a particular kind of antigen. So that's the job of a dendritic cell. Go on a fishing expedition, collect as many different samples as it can, bring that back to the T cells, because the T cell will activate. It'll get doing things once it's activated. And what's very interesting about our immune system, I'm sure everyone in this room has heard the word inflammation. And these days, it sort of gets a bad rap, mostly because we have too much inflammation in our bodies. But under normal circumstances, inflammation is absolutely essential. You trying to heal from a wound. You have cancer. Your immune system is what takes out these abnormal cells. And so it's a part of the healing process, but our immune system and these immune cells are a little bit like an all-in-one wrecking crew. They go in and they're like, where's that cancer cell? Where's that pathogen? I need to heal that wound. They want to get in, they want to get out, and that often means a lot of damage to surrounding healthy tissue. But if you're just healing a wound for a few days, that's no big deal. If you just got a few cancer cells, the immune system gets in there and takes them out. You've got a flu virus, gets in there and takes it out. Where we have a problem is when we have inflammation going on at a very low level, probably not even noticeable to a person, day in, day out, weeks, months, and years. Because what's happening during all of this time is that low-level damage is occurring to healthy tissue. And so microbiome research, if you've looked into it at all, there's a lot of it that focuses on the inflammatory process and what's happening when that's going right and what's happening when that's going wrong. And there's about a dozen or so T cells at least, but there's two in particular I want to draw your attention to. One is called the T regulatory cell, and the regulatory part hints at what that does. It quells inflammation. It regulates inflammation. And there's another kind of T cell called the Th17 cell, and it's pro-inflammatory. Because we are walking around in an environment where we might have some kind of a wound, pathogen, cancer, whatever. You need your immune system to be on the ready all the time. If you need some inflammation, great. Fire up some Th17s. Okay, the problem's solved. You need that to back down, so you're going to fire up some T regulatory cells. And as I've been showing these slides, I wonder if anyone's been sort of looking at what's going on here. You have dendritic cells that are capturing a molecular sample from particular bacteria living up here. And you have your dendritic cells that are getting another kind of antigen that's activating the pro-inflammatory cell. So what you have here is dendritic cells that rely on certain members of our microbiome to tee up a Treg cell and other members of our microbiome to activate the pro-inflammatory cells. So you can see that this is a balancing act. That's what this is, you know, ideally, most of the time, we want these things to be in balance. And this has been a huge area of research in the microbiome field. And I want to say just one more thing. I now think about my immune system very differently than I did before writing this book. I never really thought about the microbial world as being a kind of a puppeteer, if you will, of our immune system. Sure, we're all familiar with what happens if we get some kind of a pathogen in us, but how many of us have thought that it's actually the presence of microbes, because of what they teach and allow our immune system to tune into, which are just as important to have, and to have in large numbers in many diverse kinds, compared to pathogens. So this was a whole new way of thinking about what health is. Oh, it relies on an abundance and diversity of microbes in our bodies, as opposed to their absence. So when you begin to think this way, you start to look at things differently. And one question you ask is, my God, it's 2016. How has this escaped us? You know, we are in the modern world. Well, there's some very good reasons for it. It's called germ theory, because at one time, mostly what has been the bane of humanity up until very recently has been infectious diseases. We're a big, big problem. And this is a bill of mortality from 1664 here from the city of London. This is some of the earliest epidemiological work that we know of. And so you go around every week and figure out why people died, and then you chart that up, and then you can start to have some data and a record to rely on. And there's some very small numbers on here. Let's take, there's egg, 2. Not sure exactly what egg is, A-G-U-E. It's some kind of an ailment for sure. Okay, you've got some
flu, it's kind of fever, flu.
Yeah, fever, flu-y thing, right? We can all relate to that. And then you look up here, and we've got plague. Over 7,000 people in 1 week have died of the plague. This is why we have concerns about the microbial world, because there are parts of the microbial world that are not nice and friendly and warm and fuzzy, as I myself found out with the HPV, right? So this is the thing about our microbiome. We want to go skip and dance along and say, my microbiome is wonderful. And then we have to say, well, wait a minute. Sometimes there's ways in which our microbiome members are interacting with one another. You have pathogens surfing into this situation. So the microbial world is very mercurial, right? It's got this duality to it. And we humans, we like black and white. We like it all good, or we like it all bad. And the problem is that we painted the entire microbial world as evil, because that is what we had learned from germ theory. That is what epidemiologists and doctors and clinicians and everybody who's dealing with human health problems has thought about the microbial world forever and ever. However, we're going to fast forward in time, and what these two figures show is that, right post-war years, a number of infectious diseases, rheumatic fever, tuberculosis, mumps, measles, and so on, big drops in their incidence. And this is really explainable. Vaccines have done a lot to combat infectious diseases. Clean drinking water has probably done more than anything else. And then, of course, this was the advent of antibiotics. So this explained a lot of the reason that we've had such steep declines, at least in the Western world, in infectious diseases. But around the same time period, there's other diseases that are doing the opposite. There's these huge increases in the incidence. Type 1 diabetes, multiple sclerosis, Crohn's disease, asthma, and so on. And so this became a great puzzlement, to those working in this field. Why is this happening? And so there's a hypothesis about it, and you can probably guess where I'm going to go with this, right? So if you think back to those slides about the colon and the microbes that are there, and how different microbes are activating different kinds of immune cells. Well, what if we've knocked out a part of our microbiome? So you have these dendritic cells looking around for that particular antigen to tee up a particular kind of T cell or another type of immune cell. But we don't have that member in our microbiome anymore, or it's taken a hit that particular week or month. And so the hypothesis is, are we missing some of our microbes? Causality or correlation is not causality. This is where microbiome scientists, are really digging into the details to do connect the dots to prove, yes, missing microbes equals an increased incidence of chronic and autoimmune diseases. But it's looking like that's the way that things are headed. Because this is a list of various ailments that, they say 7 of 10 Americans have at least one or more chronic conditions. And these are a list of various things in their alphabetical order. So it's thought that a perturbed microbiome, an altered microbiome, is in part at the root of this. And we're not trying to say, we always like to find that smoking gun, right? Well, the microbiome is probably not the smoking gun. There never is one. Because we all have our genes, right, from our parents, plus our microbes. And we have environments that we live in. And so all of these factors are working together, microbiome, genes, and environment, to affect our health. So this is, I predict future years, Nobels are going to be awarded to microbiome researchers. We're going to be unraveling and figuring out how all of this is working. So obviously we know antibiotics can alter and change a microbiome, especially taken early in life and over a long time period. But there's something else that's far less obvious, and it has to do with this. It has to do with our diet, with what we're sending down our hatch. Now, why would this affect our microbiome? Well, just like Dave was talking about all those rhizosphere, all the microbes in the rhizosphere, the plants are pumping out sugars and proteins and all these goodies. These microbes need to eat. They need to eat to power their activities. And when they're in our ecosystem, they rely on you, on me, on everybody else who's eating food to power their activities. And what's quite interesting about the microbiome is that it has shed some light on why certain kinds of diets appear to be more beneficial for us than others. And in order to understand that, you need to understand sort of how food and different foods move through the digestive tract. Over here, this is just showing the increase in bacteria as you move down the digestive tract. There's just hardly anything in the stomach. It's a very acidic environment. There's not much that can live there at all. But as you descend the digestive tract, the acidity drops off. It really increases by the lower part of the small intestine. What's happening here is that in the small intestine, simple sugars, proteins, and fats, these are foods that are fairly easily broken down by the gastric acids in the stomach and even by our own digestive enzymes. So those types of foods, by the lower part of the small intestine, they're mostly absorbed into your bloodstream, taken off around your body to wherever they're needed. But there's some other kinds of foods that we are just miserable when it comes to breaking them down. We lack the enzymes for breaking down complex carbohydrates, the whole universe of plant foods, from a whole grain to a kale leaf to a carrot. You all know what fruits and vegetables and whole grains are, right? So this is very interesting because we don't have the enzymes to break them down, but our microbiome does. What happens when they hit the colon, when these complex carbohydrates hit the colon, where the lion's share of our microbiome is they get to work breaking that stuff down, eating it to power their own activities. And they do that through fermentation. And just as Dave talked about microbes in the rhizosphere producing metabolites, our gut microbiota, particularly those in the colon, are also churning out metabolites based on what we eat. And so when you eat a diet that's rich and abundant in plant foods, and this isn't to say that's all you should ever eat, but it is to say eat a lot of them and your microbiome becomes somewhat like a pharmacy. Your colon becomes not an onboard garbage can, it becomes an onboard medicine chest that you have the power to stock. Feed your crows, and they will stock that medicine chest. An example of exactly how our diet has changed in relation to carbohydrates in particular. We eat about the same amount of carbohydrates, somewhere around 500 grams per day, in 1910 as we did in 1997. And you can extend this out, and it hasn't changed that much. What's changed is the form of those carbohydrates. Back in the day, let's say, it's what nutritionists would call high fiber. So this is to say that the plant foods have very much remained in their whole form. And so for a grain, this is particularly important. You take a grain and you take the bran and you toss it off, or package it up and sell it by itself. You take the bran and you do something else with that. Well, all that's left is the endosperm of that seed. Endosperm is like sugar, pure sugar. It in fact is the equivalent of a placenta for a germinating plant. It is filled with energy. The implications for our microbiome are this. You're eating sugary foods, they're out of your system at the small intestine. And you've got the lion's share of your onboard, medicine chest makers down in your colon going, hey, I need some things to ferment. I need the bran, I need the germ, I need the apple, not the apple juice, and so on. And so this is how diet can really affect the microbiome. These days, we're just eating carbohydrates that are very low in fiber, and that's simply, plain and simple, it does not feed our microbiome. So we've got this sort of malnourishment going on in the ecosystem, and what we really want to be doing is properly nourishing it. And so let's put all this together: diet, immune system, and microbiome. Okay. So down the hatch comes some fiber. Okay? If you're a nutritionist, you have a lot of different names. There's a lot of different kinds of fiber by virtue of the fact that there's just a lot of different plants and plant foods in this world. Microbiota excel at fermenting that, and when they do, they kick off metabolites. These are three fairly well-known ones. This one in particular, butyrate, is very, very interesting. Microbiota has chowed down on that plant food, kicks off its waste product of butyrate, which turns out to be medicinal in us. Because, take a dendritic cell, you know what they look like from those previous slides, put one right here. The dendritic cell goes up here, catches some butyrate, takes a sample of it, comes down here, show it to a T cell. That T cell says, oh, those are my marching orders to become a T regulatory cell, the type of T cell that is the inflammation dampener. And so for Dave and I, when we were able to connect these dots from diet to what our microbiota and our colon does with that food, to churn out a compound that communicates with our immune system, I mean, wow, we were sort of like, even for people like us who realize we're embedded in nature, we were like, wow, we're really, really hand in glove with nature if, you can find the right glove and be doing all the right things. So we found this to be very, very helpful for us in thinking about how we eat. It's motivated me to change my diet. I'm a person who's got a lot of interest in my immune system, keeping it running in tip-top shape. And so this was where we landed. And it gave us some ideas, too. We began to see, and I've seeded this ground for you, that the root is the gut and the gut is the root. These are two stunning parallel universes. And if we can mimic some of the things that we know are going on in the soil in regards to soil fertility and how that figures into plant health, maybe we ought to consider that we've got sort of an inner garden in this grand ecosystem of our colon as well. And so here's just a few of the key parallels. Those exudates, they feed the microbiome in the soil. And in our case, our diet is kind of an exudate, if you will, but also some of the mucus that our colon cells produce contains exudates. And been yammering, it's these microbial metabolites, both here in our gut and here in the root. They are some fabulous compounds that contribute to the health of plants and to the health of people. In fact, it's estimated that somewhere around 40% of the compounds circulating in our blood have a microbial origin. So that kind of tells us, you know, our microbial partners are doing something, you know, either for us or to us. And there's a lot of unraveling of that and learning more about how all of that is working. And then lastly, we know in the plant world, we go into this in more detail in the book, but you know, plants and microbes are in cahoots when it comes to the plant defense system. Without microbes, you know, plants are dead in the water. There's all of this back and forth communication about who's munching on my roots now, who's going to be munching on, you know, the roots in about 5 minutes. I'm getting all these chemical signals. Okay, let's ramp this up right now. And likewise in our gut, our immunity, our defense system relies on communication and chemical signaling and all kinds of back and forth conversation, crosstalk with our microbiome. And well, I just emphasize here too, all microbes, you know, not just the pathogens. Those are for sure communicating with our immune system. The immune system's got that figured out. There's all of these other ones too that are teeing up things and tipping us off to what it is that is threatening us and what is helping us. And of course, this led us to ask ourselves, what is it that what should we be eating then? If we were trying to cultivate this inner garden, what kinds of things do we know that our microbiome might like? So we just came up with sort of very simple way of thinking about it. The Western diet, we're all familiar with that. The problem here, I'll draw your attention to the two largest arrows on this slide. We've got this one here, the Western diet, and it's really, really high in simple carbohydrates. And we know that that does nothing for down here. And the result is you get very few medicinal microbial metabolites when you're not feeding your microbiome, right? But I'm the gardener, I got to call it the inner garden diet. And it's like, don't kill yourself and never have a baby again, folks. We don't mean that, but we mean think about your whole diet. And let's see, today, have I fed my microbiome? Oh, yeah, definitely going to have vegetable-heavy dinner because I have, you know, the other stuff for breakfast or for lunch. So have some, not too many. We go into these two areas more in the book. But this guy, an abundance of complex carbohydrates, get them down your colon, microbes ferment them, you stock your medicine chest, and then you can have a cinnamon roll 2 days from whenever you did that. So it's all about balance. And I think most of us in this room probably know that in our heart of hearts and our stomach of stomachs. It's always been about